Hi everyone, this is Citizen Zeus. The moniker I use as a citizen is for Dr. Zeus Yamianis, PhD. I have a degree in philosophy of education where I explored the cultural foundations of how we learn and how we get information. And along with that, I have a biology background where I worked in physiological chemistry lab, so I'm not intimidated by scientific language. By bringing those two together, critical media literacy, the understandings and underpinnings of cultural foundations, and an understanding of science, I am able to bring you a comprehensive, integrated, critically aware, and scientifically literate understanding of many of the issues today. You can support me at citizenzeus.com, and if you want to, on my YouTube channel as well. And if you feel moved, please donate. There's a button there on the citizenzeus.com as well as my YouTube channel. Today we're going to be discussing, is there an HIV segment or an HIV-like segment in the COVID-19 virus, the SARS-CoV-2 virus? We're going to have some interesting insights into that, but to basically summarize it, yes, there appears to be an HIV-like, maybe not exactly HIV, but an HIV-like function, gain of function in this new virus, which makes it a much more able to transmit and get into human body and proliferate. But the good news is, that's the bad news. The good news is that if it was bioengineered, and I do say that that's more probable in my video called Hybridizing Harm, then they didn't do a great job of it. The insertions that they made, the HIV-like insertions in there, aren't as quote unquote good. In other words, since they escaped ostensibly from the Wuhan Institute of Virology, which I also discussed in that video, they are like a lot of Chinese goods, somewhat of a knockoff and not a very high quality one at that. Now, this is not a challenge for China to try to upgrade their ability to infect the human immune system and the human body. Please, I hope that we can all learn from this. I'm glad it was a knockoff. Thank you, China. Now let's stop doing this gain of function research, period. And many of the cloning and many of this genetic engineering have no service. They are said to be trying to predict viruses by creating them in advance so that we can have immunizations and so forth against them. The danger of these things is far greater than any kind of benefit that can come from them. The ability to outstrip our own immune system and our own ability to learn in a stepwise fashion where we ourselves, if you will, mutate or learn along with the mutating viruses and bacteria and learn to work together in this process is what it's all about. That's how nature learns. That's how nature progresses. That's how nature develops. We start messing that with some kind of Frankenstein arrogance, then we have a problem. So I am glad that this genetic engineering apparent genetic engineering to insert HIV-like or Ebola-like segments into a SARS backbone, a SARS, which is a respiratory virus, have been, let's just say, dodgy. It's now allowed us, and we can see that it's allowed us, if we maintain good health, to largely avoid some of the worst aspects of this virus. And we're going to see why that is in this. There are two things that really come up here that make it less as strong than an HIV virus. One, the protein, the shield that this, and we'll see this in an article that we're going to do, the shield of these HIV-like segments don't work as well as HIV. It allows the body to recognize, it has a little bit more hold. It doesn't protect or camouflage itself as well. And its ability to proliferate, they're called glycoproteins. We'll look into that as well. That has an issue as well. So it's able to signal its presence and not camouflage it as well. It's also not as effective in getting into cells and so forth. So we'll get into all of this. Hold on to your seats. It's a nice comprehensive. It doesn't take one side or the other. As I said before, I think this is a product of scientific arrogance more than intentional release. It was an accidental release, and I also believe it's more about scientific arrogance than it is about bioweaponry or some other thing. Not that that's any better. We have a lot of arrogant, top-down controlling efforts in our lives, and we really need to resist that through the development of bottom-up awareness that goes beyond mere conspiracy, goes beyond party identification. And that's what I'm going to be doing in this video. So let's just get me down into the corner here and jump right into it. Here's the first thing. I've looked up this notion of HIV components, and you get all over the map. You know, you've got the basic media representation. HIV did not contribute, and we will look into that. And then it says, and this is the one I really liked. Unlike HIV, coronavirus has a weak shield. That one was a nice medium one. And again, a lot of the propaganda around there's no HIV. And then there's some that actually say that uncanny similarities between the unique inserts into this SARS-CoV virus, SARS-CoV-2 virus, or this is another name for the virus, the 2019 nCoV virus. And one says, don't believe the conspiracy theories. And then you go to the next one, it says uncanny similarity. And no, it was not genetically engineered. And then it keeps going back and forth. So as I say in an article I have summarizing on my site, an investigative series into the nature, origins, and effective address of COVID-19, effective address, is that is to really look at its function and then determine what it is and where it came from. And only do that insofar as it's going to actually help us meet it in some way, engage it, not be harmed by it. So as I say on here, it says the second thing, the first thing I already noted, I do believe it was released accidentally from a lab. I don't really find evidence of more nefarious motives like bioweaponry, international intrigue, or economic sabotage. That does not make it any better. It's still killing people. The second thing I said is how the virus itself is kind of a Frankenstein creation to stitch together a whole chain of capacities that allow it to, one, transmit easily through the airs. That's the SARS backbone part. And then attach to human ACE2 receptors, which are proliferated in the lung linings and so forth, and allowing it to camouflage itself and piggyback on human cells and to cleave through cell walls and enter by basically tricking these furins, these proteins, to cleave through and then get through into cells using these human cells' own cleaving mechanisms, these furins, and then to proliferate by being able to access other cells once the virus replicates itself within a cell. So these are the ways in which you become, viral load. You get a lot of viruses in you. In this particular case, the transmissibility through the air allows it to proliferate well. But in the old SARS, it didn't have the ability to attach to cells, cleave into them, and replicate nearly as well. So instead of millions of people, like mathematical projections said, it was only a few hundred people that died from these things. And this one actually, in some sense, is no exception, as we will see. It has serious vulnerabilities, and if we just directly target those, we will find that our death rate from SARS-CoV-2 is basically as low and could be even lower than, say, a normal flu if we use the proper precautions. So it has these things, okay? It's a Frankenvirus. It has a SARS component, which allows for respiratory virulence or dangerousness, ability to create disease. Its protein spike allows it to evade human or immune defection. Its protein spike allows it to evade immune detection, attach and enter human cells. And this seems to mimic many of the actions of an HIV virus. Again, it's not exactly the same. It just mimics HIV-like. And indeed, some of the protein sequence are shared with HIV, leading many to believe that it's all planned pandemic, etc. I don't think that is as helpful. I think the easier and more direct and simple explanation is it was genetic engineering, and it did, in fact, get out of the lab. Now, what can we learn from that? We have learned a lot, not just in terms of the sloppiness and the danger of certain kinds of scientific research, but we've learned a lot. COVID has done a lot to reveal to us the control mechanisms and the facades behind political parties, behind people trying to do well for us, like immune companies, vaccination companies, and so forth. It's really, and the unfairness of people bailing out huge corporations that are already profitable and then not getting checks themselves. So in many ways, I do thank the COVID-19 virus for revealing in a way that no other event, I think, could have, the mechanisms of control, the mechanisms of exploitation in this world, and the need for us to really take control of our own health. So that's the real upside to this. We got a message, instead of being that, what they say, the frog with the boiling water, boiling to such a gradual degree we didn't actually know that we were being cooked alive. This COVID smacked us. It turned up the heat real quick and created a whole bunch of responses, which it revealed. So this job here is to really zone in on what we can learn from the HIV-like function of this virus and what it teaches us about health and what it teaches us how to respond well to this particular virus, but also viruses in general. So let's go ahead and go to the next slide. Now, all of these, as you can see, you can pause at any time. You can pause at any time and get a snapshot of these sources, but I will have them in the description below. So you can tap onto any one of these in the description below. And again, if you feel moved, please pass it along. Please subscribe to my channel and donate too if you would like to help to support this kind of research. I spend many hours doing this. This was a very good summary article. It was neither pro-HIV or anti-HIV, and it says it right. It says, and this is actually from China. It's the South China Morning Post, well written by this Stephen Chen. He said the coronavirus is far more likely than SARS to bond to human cells due to an HIV-like mutation. And this is according to scientists. So here are the relevant paragraphs here. The new coronavirus has an HIV-like mutation. That means its ability to bind with human cells could be up to 1,000 times as strong as the SARS virus, according to new research by scientists in China and Europe. 1,000 times more likely. Now, is that just going to actually just come out of nowhere and just naturally happen 1,000 times more? Most people, most thinking people would say no. It seems to be more intentional. Again, here's the good news. Unlike all the movies saying that we are creating AI or something that's going to take over the world, nature is a little bit wiser. When we do mess with nature, nature tends to, within a 4 billion plus billion-year-old wind, tends to kick out our paltry attempts to play God. And I'm hoping this is the case with this one. If this was artificially manipulated and inserted into this virus, through mutation, through our own learning to deal with this, and there are many, many good news around that. Children, almost none of them between 0 and 18 are dying from this, far lower than most other diseases, even common diseases. So by being given a certain amount of grace here, we're actually going to be able to learn how to handle pandemics. We're going to learn how to handle our health. In this case, we're going to learn what the nature of this particular virus is and the dangers of human manipulation. So scientists showed, here we are right here, that SARS, severe acute respiratory syndrome, entered the human body by binding with a receptor protein called ACE2 on a cell membrane, okay? And some early studies suggested whatever. But as we move down, but the ACE2 protein does not exist in large quantity in healthy people. This is very important. This is one thing they don't tell you because they might want to make it look like, oh my gosh, the viruses are just going out to get us. The reason why ACE2 protein does not exist in large quantities in healthy people is its purpose is to basically scavenge and signal when cells are damaged. When you're a healthy person, you're not smoking cigarettes, you're not eating junk food, you're not getting involved in electronic pollution and hanging out indoors and not getting your vitamin D. When that happens, you create oxidative stress in your body and you create a lot of damage to your cells. So these ACE2 get produced in higher quantities to help signal and create a system of redressing that damage in your body. Now the SARS-CoV-2 hijacks that process, and something that is supposed to, in some sense, tamp down your immune system, okay, and supposed to help create a smoothing effect or an ability to signal and bring your body to bear on problematic areas, including damaged tissue, not just external invaders. Then all of a sudden, when it hijacks that, it's creating this weird confusion in your immune system. And we see this, as I showed in my other videos, as a cytokine storm or a weird overwhelming autoimmune response that begins to just rip the body apart. And then most people who have died, they have diabetes, they have heart problems, they have underlying conditions because they have those extra ACE2 receptors. They're already in a weakened condition. And this new virus is able to trick it to create an overwhelming immune response, which creates multiple organ inflammatory syndrome in very few kids, but certainly in adults, and this massive clotting that just basically freezes up the lungs, stops blood flow in capillaries, and makes it very, very difficult for oxygen to get into the body. That's how it works, scientifically or just sort of logically. And because it has that respiratory ability to enter in, and combined with an HIV ability to get into the blood and really mess with the immune system, you kind of have a strong 1-2 punch. But because it's not as good as, combined as it is separate, these two functions provide an entry point for us to say, wait, we don't have to be intimidated by this virus. We can learn from it. And we're going to find out exactly how in these upcoming few articles. I'm doing a shorter one than the longer 45-minute and sort of. I'm only using about 5 or 6 articles here. Here's how this SARS-CoV-2 virus is like HIV. Okay, now don't say it's the same, because then you give people who are skeptics the ability to say, it had 1 or 2 proteins off. It wasn't the exact same thing. Or if you say it was, a military or bioweapon, they go, oh, you're just conspiracy. It doesn't have to be conspiracy, and it doesn't have to be exactly like an HIV to be a mutation that works like it and has a lot of the same problems. So other highly contagious viruses, including HIV and Ebola, target an enzyme called furin. This is where this is different, which works as a protein activator in the human body. So many proteins are inactive or dormant when they are produced and have to be cut at specific points to activate them. Basically, the HIV component in this virus helps trick that furin into activating in such a way that allows this SARS-CoV-2 virus not only to attach to the cell, but to cleave and enter into the cell. That's what makes it different and much, 1,000 times or 100 times worse than your basic SARS. The basic SARS doesn't have that. The SARS backbone in this allows it to be transmitted, but it's an HIV-like component that allows it to cleave, enter into the cells, and proliferate better. That's why it's a more dangerous one and a more deadly one. There's only a few hundred people that died from SARS. Now we have up to 400,000 people dying from this one, and the numbers are still piling up. So this finding suggests that the 2019 nCoV, which is the SARS-CoV-2 virus, may be significantly different from the SARS coronavirus in the infection pathway. And the virus may be using the packing mechanisms of other viruses like HIV. Here's the relevant thing that I've highlighted. The virus uses the outreaching spike protein to hook onto a host cell in your body or in somebody else's, but normally this protein is inactive. That's where the SARS got hung up. The cleavage site structure's job is to trick the human furin protein, that's what we produce, so it will cut and activate the spike protein and cause a direct fusion of the viral and cellular membranes. So when the old SARS virus couldn't do that very effectively, couldn't enter very effectively, this one does. It tricks our own bodies into creating a cut or an entry point, and then it fuses and then enters into our cells. Kind of, scary, but not scary, because we're finding that there are extraordinary ways to prevent them from even attaching. And I've gone ahead and talked about those in other videos. I encourage you to look at some of them, especially those that attach to these ACE2 sites, and prevent the virus from replicating, attaching, and getting into your cells. I have one on cannabinoids here coming up. But anyway, let's look more into this mystery here. This is an article by Jean-Claude Perez. And again, this man is not an epidemiologist or a virologist. He's a retired interdisciplinary researcher. But what he does seem to have is an understanding of engineering. And he is not intimidated by these by this language, the scientific language. His conclusion was this. The main result is that it was, in his conclusion, a synthetic genome by identifying these four insertions that were very much like an HIV protein sequences. What he talks about, and I think this is important, is this part here. All this is remarkable, the fact that these insertions are very HIV-like and bears the mark of a desire for organization of a human nature, logic, and symmetries. When you're looking at something, as I said in my other video, you have to look at the motive, means, and opportunity, right? The motive here, in many ways, is scientific arrogance, patents, developing something and then developing a vaccine to it, being this big star of genetic manipulation in the scientific world, you know, just very, very ego-oriented stuff, okay? So that's oftentimes the motive. The means are these viral laboratories in which these kinds of methods have been developed. You will find no article that says it absolutely could not have been done by a genetic lab. It just says it wasn't done by that. And then they try to offer evidence as, well, you know, scientific, techniques usually use this thing and it wasn't involved in the lab. But I've read comprehensive papers that show not only can this be done in a lab, not only can it be hidden, and that is the evidence of human manipulation, you know, and insertion into this. Not only can it be hidden, but it can be done pretty easily, even by graduate students. The technology is not that high now because the instrumentation has been so advanced. So it could be done. And the logics and symmetries of this, doing this, and the way it's being done seems to suggest human intervention. The fact that it is, and I will talk about this later, but we'll move to the next article because it does talk about this. This is an article from Zero Hedge. It does a pretty good job. It says it contains HIV insertions. Again, I would caution you to say HIV-like insertions because they're not exactly identical, but they are very close. Okay. Stoking fears over artificially created bioweapon. Again, I think it's more about scientific arrogance than a bioweapon, but here's the main point here. It's a good article. You can read through it. Takes apart the paper in clear, you know, easier to read terms, the scientific paper, but here's the relevant line. Why do the authors think the virus may be man-made? Because when looking at the above insertions, which are not present in any of the closest coronavirus families, it is quite unlikely for a virus to have acquired such unique insertions naturally in a short duration of time. Instead, they can be found in cell identification and membrane-binding proteins located in the HIV genome. These don't normally go together, HIV and the stars, okay? And for those kind of mutations to sort of find their way through natural processes, it would likely take a long time. But I've read papers, and I'm going to go over one which is so comprehensive, that you can do it in a lab. You can create these mutations in a speeded up form by passing them through a membrane over and over and over again. So those kinds of things allow us, even when things are many thousands of protein strands off, to literally create mutations within a fairly short amount of time that can alter significantly the makeup of a particular virus so that we're manipulating, or bacteria that we're manipulating. So again, I think the time factor begins to lend itself to this is an HIV-like insertion that was man-made, not nature-made. Now you get articles like this, HIV-1 did not contribute, a bold statement to the SARS-CoV-2, or also called the 2019 nCoV genome. Again, look at the language here. HIV-1 did not contribute. They didn't say HIV-like. See, they're trying to distract you with the way that they use language. And they try to make it a very bold statement so that your attention is focused, oh, it's not HIV. Who cares if it's not HIV? If you extracted protein strands from an HIV with the same gain of function and you drop them down into a SARS backbone, then it really doesn't matter if it's exactly the same as HIV. You have made a gain of function that's very much like it. As we will see, it's not exactly like it, and thank heavens for that. It's not as effective as an HIV virus in doing these sorts of things, and we'll investigate that. But thank heavens they didn't do a very good job. I've mentioned this again. It's like this research came from a lab. Fort Detrick linked up with the University of North Carolina. the premier genetic engineer around this. I can't remember his name, is Sar something or other. What was his name? Garvik or something like that. Get into a competition with China. And I think it had more to do with scientific competition, trying to identify new viruses, trying to manipulate them, and trying to develop new capacities. And they really just got ahead of themselves, and they really got too arrogant and too sloppy, and they created something that could do a lot of damage. So look at the language here. Lack of the definite origin has led to speculation of the 2019 nCoV could be derived from genetic manipulation or even for the purposes of use of a bioweapon. This notion has been fully debunked in the media. What kind of scientific paper would say this says this notion has been debunked in the media? Instead of giving you an actual scientifically clear and concise statement that says, this could not be because of this. There is no, in my research, debunking of this. The best paper that I saw was this one here. It is the most extensive. It's written by somebody who has a knowledge in the field. He's a young guy, and he basically, this is the most comprehensive paper I said. I encourage anyone who's a bit of a science nerd to look into this. He does a pretty good job of stating right up front. But the fact is this, and I'm just going to read it. This paper is scientifically literate. It gets into the details, and here's what it says. Again, it says, lab-made SARS-CoV-2 genealogy through the lens of gain-of-function research. It's targeting that gain of function. That is your smoking gun. If that gain of function doesn't look like it come about in nature, except through long evolution, and all of a sudden it's here, and you know that in a lab, they can accelerate evolution in the process of, and they can genetically insert things that would not normally have found their way there, then you have to conclude it is likely the result of a lab or artificial intervention than a natural one. They have never produced very good rationale, much less proof that this is of natural origin. They keep trying to say there isn't clear-cut evidence of it being in a lab. That's because you can just destroy your research. That's because you can just go ahead and hide where all this stuff came from. Logic, people, critical thinking, critical media literacy, and this guy does a great job. So here he says, if you hear anyone claim we know the virus didn't come from a lab, that's an arrogant statement. No one can prove that. Don't buy it. It may well have. Labs around the globe have been creating synthetic viruses like COVID-2 for years, and no, its genome would not necessarily contain the hallmarks of human manipulation. Modern genetic engineering tools permit cutting and pasting genomic fragments without leaving a trace. It can be done quickly, too. It took a Swiss team less than a month to create a synthetic clone of COVID-2. Less than a month. And we keep on going down to, he thought it was nonsense at first. Then when he began to research, he said, wait a minute, okay? So he said, all right, yeah, I'm going to go ahead and look into it. And he said, the one thing we can know is that the COVID-2 is an obvious chimera, okay? Though not necessarily a lab-made one. A chimera is one that, takes basically backbone of a particular thing and creates a gain of function that allows it to be more effective. In our case, in infecting and taking root in the human being instead of just from others. So so as he says here, COVID-2 is an obvious chimera, which is based on the ancestral bat strain. That's the one found in China. We already have identified that. And it's 96% similar to this SARS-CoV-2 virus, okay? 96%, by far the most similar of any out there, in which the receptor binding motif, that is this spike protein, is replaced by the RBM of a pangolin. A pangolin is a different animal. And in addition, a small but very special stretch of those 4 amino acids is inserted, which creates a furin cleavage site that, as virologists have previously established, significantly expands the repertoire of the virus in terms of what cells it can penetrate. So, this has all been established. You can't prove outright. And I'm going to go over this paper and just using this paper to get you into the intrigue and likely scenario of scientific competition arrogance, and opportunism that gave rise to this disaster, and unlikely a bioweapon. But let's move on to the next one. We have here, and this is where there's some grace. Unlike the HIV, the coronavirus has a weak shield, though it has an HIV-like mutation and function, okay, that could easily have been inserted, and I think logic suggests it was inserted by these labs. It doesn't function as well as HIV, thank heavens. And in two particular cases, in two particular points is very important. This is what this one says, and it's actually well written. It's in Forbes magazine, but it's one of the best written articles on this. It says, although many viruses may not need glycans for their proteins to work properly, glycoproteins, these are sugar proteins, okay, are so common in their hosts that sugars are often added to form a glycan shield around a viral protein to help hide it from the body's defenses. So what the virus does, and the SARS-CoV does this too, is it creates a sugar shield around its protein so that the body won't recognize that it's an invader. Sugars are, very common food. They're especially in their simple forms, they directly help run the energy centers of the cells. Okay, so your body doesn't want to be allergic to those or to go against them. Otherwise, you would starve to death. Okay, so how does the SARS-CoV-2 compare to HIV, which is infamous for its ability to evade and infiltrate the immune system? Well, the two have similar numbers of these sites, these glycosylation sites. And the SARS-CoV-2 spike is larger than that of HIV, the coronavirus's glycan shield is actually less dense and the sugars have a different chemical composition. So its protection, its camouflage is less dense, meaning it's less tricky, it's less effective. It doesn't look as much like a sugar. And the sugars have a different chemical composition, which allows the body to more easily recognize that it is invader. It's not as good at camouflaging the COVID-19 virus, and thank heavens for that. And you can see that in these pictures. Glycan shields over here, much more numerous and tightly packed, dense. Glycan shields here, not so much. Okay, thank heavens for that. Our bodies are able to recognize the SARS-CoV-2 virus better than the HIV. Interestingly enough, though, both of them do work very similarly. They attack the immune system. We've seen that in the SARS-CoV with this odd inflammatory, overreactive, confused immune response where the body's attacking itself, much like the HIV, and you get underlying weaknesses and conditions brought forward, pneumonias, organ problems, etc. And that's why, again, diabetes and other diseases are much more implicated because they're already damaged. They're already producing these ACE2 receptors. There's a lot more places for these things to glom onto, replicate, and make more problems and mischief with your immune system and your blood system. When I did my video on, is this a respiratory disease or a blood nerve disease? I said it's basically both, and you can read that again in my article, a summary article on this. So, and this is the other part of it. It says, besides density, the distribution of glycans can also differ from HIV and SARS-CoV-2 between them. In HIV, the sugars surrounding the spike's receptor binding domain block access to antibodies. But in the SARS-CoV-2, the receptor binding domain is exposed, which presents a potential weak spot. So, it's shielding the receptor binding domain. It's kind of shielding the docking. Got docking doors there. The other one is more open. When that happens, then it's easier. It's not as shielded in that region. So it's more, as it says here, vulnerable to antibody neutralization. Antibodies can neutralize it better. As a result, for those two things, it's less shielded, it has less proteins, or sugars, less coverage of sugars, which allows our body to detect it and respond quicker. Let's go on to the next one. I just brought this up to show you that much like HIV, what we've seen as presented as the symptoms of SARS-CoV-2, cough, right? This dyspnea, oftentimes presented in pneumonia, chest pain, fever, abnormal lung exam, hydrogen saturation. They are all exactly the major symptoms and presentations of the SARS-CoV-2 virus, this COVID-19 virus. They're presenting, but the underlying issue is a blood issue and an immune issue. And if we move on here, will note that this gets to such an extreme in HIV. This is HIV. This is another one of the issues that happens in HIV. It's called PCP, pneumocystis, which is an actual parasite here, okay? And it's present in most people's body. But when an underlying illness that disables and confuses the immune system, oftentimes to either not effectively attack a virus, or in this case, because it's camouflaging itself within cells designed to signal and help our immune response, it can create an overwhelming immune response because the system is too confused, where the immune system can actually end up shutting our body and killing us, much like an autoimmune disease. And in another video, I talked about the autoimmune-like aspects of this disease. So it can cause disease at any time the immune system becomes severely damaged. So it's not that the COVID-19 virus necessarily kills you. A lot of people complaining that there are too many deaths talking about the COVID-19 killing people. But it becomes literally the straw that breaks the camel's back. Obviously, it's not the only factor. It's called comorbidities. It's underlying health factors, but it is the thing that pushes you over the edge. I don't think it's unfair to say that that's what caused the actual, at least immediate death. The underlying conditions are what basically contributed to the foundation of the death. And that person probably would have lasted longer had it's a COVID-19 virus not come along, but probably would not nearly have lasted as long as a healthy person in terms of longevity and life. I just stuck this in here to really begin to point in brief terms to the kinds of things we need to look at if we really want to combat this disease. It will not be a vaccine. The vaccine will take far too long. By that time, the second or third wave will already be blowing through our population. Really what we have to do is diet, exercise, in this case, vegetarianism, I would even say veganism, the thing that limits inflammatory foods in your diet, whole food, plant-based diets, lower oxidative stress. Lower oxidative stress means lower ACE2 receptors, lower damage to cells. And when that happens, not only is your overall system in better shape, but the actual receptors and cells and proteins that these SARS-CoV-2 viruses go to, albeit also a lot less. That's why you're finding healthy people are largely not dying from this. And kids, almost none of them are dying. So that's an important thing to look at. As I said in this article, again, this is the article I'm attaching here from Citizen Zeus, the investigative series that I'm doing. The central theme here is that courage, clarity, knowledge, and care for self and others is the way forward. The central practice must be excellent, comprehensive health maintenance and development, committing to plant-based whole food diets, exercises, sleep, deep nose breathing, hydration, etc., and committing away from electronic pollutions, toxins, junk foods, chronic stress, etc., all of which have been discussed in my videos. We know that the SARS-CoV-2 loves ACE2 receptors, and there is more ACE2 produced in unhealthy people in order to help handle injured cells as well as warn of potential invaders. So by eliminating the need for ACE2 through good health, we largely eliminate the body's foothold for SARS-CoV-2 to attach, break into, and replicate within our cells. If there was any epidemic that was more dependent on the literal choices we make day to day, getting out in the sun and getting our vitamin D, exercising, serious sleep, hydration, deep nose breathing and yogic breathing, all these brought together, homeopathics, as well as some maintenance of drugs like cysteine, we'll talk about cannabinoids, even nicotine has been shown to have an effect, nitric oxide from nose breathing have an effect to prevent the attachment into those ACE2 receptors. So again, it's not something that's just going to come out and get you. If you take this knowledge and you implement it in your life, that is the best protection you have. We know that certain things prevent, we have lockdowns and all those sorts of things, prevent you from getting the initial exposure to it. But given the fact that we might all be exposed to it, we need to have that underlying health to deal with it. And if we do, we're going to be in very, very good shape generally. We would need to have really, really bad underlying conditions for the most part. And we would need to have some kind of genetic predisposition. And, that happens with any disease. So this particular one actually gives us almost a recipe for how to handle it. And if we pay attention and we have the discipline and the respect of what this is showing us we need to do individually and collectively to bring ourselves into a better alignment with our health and better alignment with one another. So that's really the issue here. I just like to send this off on a positive note. This virus has done so much to reveal actual need for health, actual need to work together as communities, actual need to know not to trust some white hat from on above with our health, some wonder drug or some wonder vaccination. We literally can handle this from the ground up, from the bottom up, by really engaging these kinds of practices. And these practices are not just homespun, remedies from naturopaths and so forth. There is a lot, as you will see in my videos, if you look at them, there's a lot of indication in scientific journals that these things have effective ways to address this COVID-19 virus. So again, I really encourage you to do your research. This is not to be construed as medical advice. I am not a doctor. And even if I was, I wouldn't simply say, you follow what I say. I would say, you do the research, make this part of your research, use your own logic, talk to many people, try some of these things, see if it's going to improve your overall health, regardless of whether you get this virus or not, and see if there is a benefit here. By moving forward, by taking on these challenges and proactively understanding in a critical and creative way what it is we're facing, we become empowered on a whole lot of levels and our risk level goes way down. We don't need to look into the scary theories. We don't need to look at even the scary studies. What we need to do is find out what works, what doesn't, and why, and begin to bring those together to a comprehensive package that allow you and me to be healthier and happier and heartier when we're working with one another and when we're engaging our lives. So again, thanks for watching. Appreciate your attention. Please subscribe to the channel. Please share it with others. Please donate if you feel the need or if you feel the desire on my YouTube channel or on my homepage at citizenzeus.com. And let me know. You can comment and reach me through that site at zeus@citizenzeus.com. And I'm open to other requests for research, other information. So until next time, this is Zeus as Citizen Zeus signing off. Thanks.